Hello everyone, this is MMBS111, Introductory Microbiology. This is our final mini lecture for Chapter 12, and in this mini lecture we are going to be discussing the details of the second line of defense. There is a lot of information in section 12.3 and many things that you really need to know and understand. First, you need to be able to list the four major categories of non-specific immunity. Outline the steps in phagocytosis and the cells involved. Outline the steps in inflammation and name the classic signs and symptoms. Name the major types of inflammatory mediators and other cytokines. Define fever and discuss how it helps defend the body. Name some of the side effects of fever. Describe the role of interferon proteins. Explain the cascade reaction of the complement pathway. And finally, explain what antimicrobial peptides are. So, lots to learn. Let's get started. So, the second line of defense includes four non-specific defenses. And that means that these defense mechanisms do not recognize specific organisms. They are non-specific, very general. Okay. The first is phagocytosis, inflammation, fever, and antimicrobial proteins. So we'll be discussing these four generalized defense mechanisms in this section. Phagocytosis literally means eating cell. The general activities of phagocytosis are first to survey the tissue compartments and discover any microbes. So our phagocytes, they are going to possess specific receptors that are going to help them feel around in the tissues and recognize and distinguish foreign bodies from self cells. Then they're going to ingest those materials. So if the phagocyte recognizes a cell or some kind of particulate matter that is not self, it's going to ingest it, and then if phagocytes possess a number of chemicals that will help to break down and eliminate those materials. The other thing that phagocytes are going to do is they are going to extract immunogenic information from these foreign bodies, and they're going to display them so that they can be used to recognize those structures in the future. We have three types of phagocytes: neutrophils, monocytes, and macrophages. These are our general phagocytic cells. Neutrophils are general purpose phagocytes. They react early in inflammation. High neutrophil count in the blood is a common sign of a bacterial infection. They are also the primary component of pus, and they're typically dead neutrophils by the time they're in pus. Neutrophils do not have a long lifespan, about 3 days, and they are one of the most common cells found in the blood aside from erythrocytes or red blood cells. Monocytes are transformed into macrophages after they migrate out of the blood and into the tissues. So monocytes are phagocytic cells, and they're going to act in the blood. Once they leave the blood, then they become a macrophage. And as a macrophage, they change a little bit. They become larger. They also develop lysozymes, and those lysozymes are used to break down those structures that they engulf. So the mechanism of phagocytosis, it's the physical process of engulfment. So phagocytes act very much like amoebas. They have sort of a very fluid kind of structure. They are able to sort of create these pseudopods that then engulf foreign material. Phagocytosis can be an isolated event, so you might have a single foreign body and a single neutrophil or single macrophage that's going to engulf that foreign body, or phagocytosis could be an orchestrated event that is induced by the process of inflammation. There are several sort of events that are components of phagocytosis. First, we have chemotaxis. So we've discussed chemotaxis before when we're talking about the movement of microbes and how they either find or avoid certain things. Chemotaxis is also the process that attracts phagocytic cells to sites of infection. So there are going to be certain inflammatory cytokines and chemicals that are going to be released that will attract these phagocytic cells to those sites. Once there, the phagocyte is going to recognize that foreign body as foreign and engulf that structure, so it's going to ingest it. Then we're going to have phagolysozyme formation. So phagolysozyme is a chemical that is produced by phagocytic cells inside the cytoplasm. Those lysozymes are going to join with the vacuole inside the macrophage or the neutrophil that contains that foreign body. The lysozyme is going to be released by the lysosome into that vacuole and begin to break down that structure. Once it's destroyed, then the remaining substance will be excreted. Okay, so our phagocytic cells are going to recognize these pathogen-associated molecular patterns, or PAMPs, on our pathogens. These are sort of red flags for our phagocytes that this structure, this cell, is not self. The bacterial PAMPs are typically either peptidoglycan or lipopolysaccharide, both components of the cell wall of gram-positive or gram-negative organisms. Viral PAMPs typically are double-stranded RNA molecules that our phagocytes are going to recognize. Our phagocytes, as we've discussed in section 1, possess these PRRs, or pattern recognition receptors. They're going to recognize and bind to those PAMPs on our microorganisms. And the unique thing about these phagocytes is that they always possess these PRRs, these receptors. Whether or not they have seen a pathogen before that expresses these PAMPs, they always possess these receptors. Inflammasomes are PRRs that are found within the cytoplasm of our phagocytic cells, and they recognize those PAMPs of our pathogens once they have been engulfed. So once they've been brought into or ingested by the cell, then these receptors in the cytoplasm are going to recognize those. And then recognition by the PRRs in the cytoplasm of the PAMPs is going to lead to the release of signals that are going to initiate and regulate inflammation. And we'll talk more about these cytokines and chemical signals a little bit later in this presentation. So this image is from your text. This is a really good model that breaks down the steps in phagocytosis. So you can see we have chemotaxis happening. It's attracting our phagocytes to the site of location. The phagocytes are going to use those PRRs to recognize the PAMPs on our pathogens. They're going to adhere to those receptors. Then they will be engulfed or ingested by the cell. And then that's going to initiate or stimulate those lysosomes containing lysozyme to then merge with that vacuole that contains our pathogenic organisms. There are also a number of other enzymes that are going to be working here. DNase, RNase, proteases, and peroxidases. And these are all chemical molecules that are going to break down different components of these organisms. And then once the organisms are destroyed, then that vacuole will merge with the cell membrane of the phagocyte, and it will be released into that extracellular space. So make sure that you review this diagram or table in your text and be familiar with the steps and the components involved in each of those steps. So the next thing we're going to talk about is the inflammatory response. And the inflammatory response includes a number of steps that we'll go through. And there are a number of classic signs and symptoms that are associated with the inflammatory response. So the first is rubor. Rubor is the redness that is caused by the increased circulation and vasodilation that is associated with the injured tissue. Then we have Kaler. This is the warmth at the site that is caused by the heat that's given off as blood flow increases to that location. Tumor is the swelling that is caused by the fluid that is escaping into the tissues. And then finally is dolor, and that's the pain that is caused by the stimulation of nerve endings at the infection site. There are several factors that will elicit inflammation. That can be trauma that's caused by an infection. It could be tissue injury or necrosis that's due to physical or chemical agents. And it can also be the result of specific immune reactions. The inflammatory response can be local or systemic. So if you get a cut, generally the inflammatory response is going to be local. If the infection spreads to other parts of the body, then the inflammatory response will be systemic. The inflammatory response maintains stability, and it is able to restore itself back to homeostasis after injury. It also has the potential to cause tissue injury, destruction, and disease. And microorganisms take advantage of this. They use the inflammatory response to actually cause and elicit more destruction. The chief functions of inflammation are to mobilize and attract immune components to the site of injury. So that is why there's vasodilation and increased blood flow to the sites of injury. We can get more immune cells to the site of the infection if there's more blood flow to the site. It is also functions to repair tissue damage and localize and clear away any harmful substances, and then also destroys microbes and blocks their further invasion. Cytokines are inflammatory mediators. These are chemicals that are associated with the inflammatory response, and they help to regulate, stimulate, suppress, and control many aspects of cell development, inflammation, and immunity. They are produced by a number of different cells, so monocytes, macrophages, lymphocytes, fibroblasts, mast cells, platelets, and endothelial cells all produce different cytokines depending on the function of that chemical. And as we mentioned before, the effects can be local or systemic. They can be short-term or long-lasting, specific or non-specific, protective or pathologic. So now we're going to talk about the stages of inflammation. There are 4 stages associated with the inflammatory response. These are predictable series of steps. They can last anywhere from a few minutes to a few hours or days. Once the initial injury has occurred, then there's a chain reaction of steps and activities that are going to take place to address the injury and potential infection. Step 1 in the inflammatory response is the injury and the immediate reactions. We're going to have chemical mediators that will be released by cells at that location. They might be mast cells or some other white blood cells that happen to be in that location. And they're going to release these chemicals that are going to attract other white blood cells to the area of injury. Initially, the blood vessels are going to constrict. This will help to reduce blood loss and help protect the tissue that's been damaged. The second step in the inflammatory response are the vascular reactions. This is where we're going to experience vasodilation. We're going to get more blood flow to the site of infection. We're going to get an influx of white blood cells, particularly neutrophils. And then step 3 is the edema and pus formation. So we're going to have fluid that's going to be escaping from the cells in the damaged tissue. That's going to lead to some swelling. Also, the increased blood flow is going to contribute to the swelling. And then finally, we're going to reach step 4, where we have resolution of the injury. We might have scar formation. The white blood cells in the tissues are going to leave that site. They're going to return to the blood vessels and be transported to other parts of the body. Make sure that you review this table, that you're familiar with the steps and the general process of what is happening during each step of the inflammatory response. So there are some benefits to these symptoms that we experience during inflammation. During edema, there is an influx of fluid, and that fluid influx helps to dilute any toxic substances that may be released or produced by those microorganisms. Fibrin also is a component of that fluid, and that clot that is formed can trap microbes at the location and prevent them from spreading to other parts of the body. And then neutrophils aggregate in this fluid influx at the site where inflammation occurs. And neutrophils are our primary white blood cells that are going to contribute to phagocytosis and destroying those microorganisms. Pus we get many times at the site of an injury. This is the accumulation of a whitish mass of cells, cellular debris, and bacteria. It's also it also includes neutrophils, as we said before. Many of them are dead neutrophils. And then this term pyogenic is a term that is used to describe bacteria that actually stimulate the formation of pus. So pyogenic organisms possess some chemical substances and markers that actually attract a large influx of neutrophils to the location, which contributes to a lot of pus. Fever is an adjunct. It's a sort of, it's associated with inflammation. Fever is an abnormally elevated body temperature. It is nearly a universal symptom of infection, and it can also be associated with a number of allergies or cancers, sometimes some organic illnesses. We describe fever based on a range of temperature. So a low-grade fever is anywhere from 100 to 101 degrees Fahrenheit. A high-grade fever is anywhere from 104 to 106 degrees Fahrenheit. There are a number of substances that stimulate fever. We call them pyrogens. These substances reset the hypothalamic thermostat. So our hypothalamus is what regulates body temperature. And these substances reset that body temperature at a higher setting. Fever is actually beneficial in the immune response. It helps to inhibit the multiplication of temperature-sensitive microorganisms. There are a number of organisms that don't function well at higher temperatures, like the poliovirus or cold viruses. It also impedes the nutrition of bacteria. Higher temperatures reduces iron availability within the blood. It also increases metabolism and stimulates immune reactions within the body. It also speeds up hematopoiesis, phagocytosis, and other specific immune reactions. So there has been this debate for quite some time about whether fevers should be treated. We've already discussed that fever is beneficial in the immune response, and sometimes it can be difficult to decide whether or not to treat a fever because there are side effects to fever, especially fevers that are high and prolonged over several days. So in general, any slight to moderate fever is in a healthy individual is not treated. It's just left and allowed to run its course in the hopes that it assists the immune response. Any fevers that are particularly high or have been prolonged over a number of days are generally treated because they can lead to other issues like cardiovascular disease and seizures and respiratory diseases. Finally, we're going to discuss antimicrobial proteins. There are a number of proteins that are released and produced by the body that have antimicrobial effects. The first is interferon, and interferon is a small protein. It's produced by certain white blood cells and some tissue cells, and it's involved in defenses against microbes, and is also involved in immune regulation and communication between cells and tissues. There are 3 types of interferon. Interferon alpha and beta are produced by lymphocytes, fibroblasts, and macrophages, and this interferon stimulates phagocytes. Interferon gamma is produced by T cells, and it is involved in regulating macrophages and T and B cells. All 3 of these interferons can inhibit the expression of cancer genes, and they have tumor suppressor effects as well. So looking at just the general characteristics of interferon, this diagram is from your text, and it illustrates the effects of interferon in a viral infection. So we can have a virus or another microbe, it doesn't have to be a virus, that is going to bind to those receptors on a host cell. And that initial binding is going to signal to the cell to produce interferon. These interferon molecules are then secreted by that cell into the extracellular space, and they're going to bind to other nearby cells in that tissue. And the binding of those interferon molecules is going to stimulate in nearby cells the production of proteins that are going to inhibit viral multiplication or inhibit the effects of other microbes. So they might degrade viral DNA or prevent the translation of viral proteins. Interferons are not microbe specific, so you don't get different interferons produced for a virus versus a bacteria, or an enveloped virus versus a non-enveloped virus. The second type of antimicrobial proteins are complement. And complement consists of 26 blood proteins that work together to destroy bacteria and some viruses. Complement involves a cascade reaction. So you have a sequential set of reactions that cascade, kind of like dominoes in sequence. The first substance in the series activates the next substance, which activates the next substance, and that continues until the reaction is completed. There are 3 different pathways, and each one is distinguished by how the pathway is activated, but all 3 have the same end result. This is a diagram from your text, and it sort of simplifies the overall stages in the complement cascade. So the first step in this cascade is initiation, and this is the initiation of certain complement components, either C1 or C3, and they're going to bind to an initiator on a foreign cell. Then that's going to lead to amplification and the proceeding of this cascade. So once C1 or C3 are activated, that leads to complement 5 or C5 being cleaved and bound to the membrane of that foreign cell. The next step is polymerization. The C5 compound then becomes the site for the assembly of an attack on the membrane of this organism. And the membrane attack involves C5 through C9. So C6, 7, and 8 all together are going to form this complex. And that complex puts holes or small pores or openings into the membrane of this microorganism. And that leads to the lysis of that cell. Make sure that you are familiar with the complement cascade and the steps in this process. Our final antimicrobial protein are the antimicrobial peptides, and these are very short chain proteins, 15 to 20 amino acids, and these proteins actually insert themselves into the membranes of prokaryotic cells. And when they do that, it's somewhat similar to what happens with the complement cascade. You get pores or holes that form within the membrane of those cells, and that leads to lysis. The cell loses control over the selective permeability of the membrane. Substances leak out, substances leak in, and the cell ruptures. There are 3 examples of these antimicrobial peptides: defensin, meganins, and protegrins. And that is the end of section 12.3. These are our checking in questions. Make sure that you go through these questions, that you can answer them appropriately. Also make sure that you go back to the beginning of the presentation and review the learning objectives for this section. Here's our critical thinking question. So the human immune system includes a number of redundant cells and processes. What would be the purpose of this? So think about what you've learned so far about microorganisms and how the body responds to infection. Why would the body have a number of redundant processes in order to address infection? Once you come up with a good response, go to the next slide and check your answer for that explanation, and hopefully this video was helpful.